So for next week we do Lab 3. So you need to do the homework packet Lab 3. And I'll add it to this module and connect in case you guys lose your copy. If you're ever absent, I make enough copies for as many students as we have enrolled at that time. If you miss lab and you don't want to print from Canvas and you happen to be on campus, you can go to my office. On my office door there's a folder that has folders within it for each one of the labs. So all my extra homeworks or handouts I put in those folders, so you can swing by even if I'm not there and pick one up. Okay, so you'll notice that it says to watch some videos, right? So these are videos of me actually doing what you guys are going to do next week. And I'm going to still demo it in front of the class, but the more exposure you guys have to this stuff, the better it is for you. Okay, and there's actually a question that I ask here. I actually make a mistake in this demonstration. So see if you can, after you've read up on the homework, right, and what it is we're actually doing, what aseptic technique is, watch this video and see if you can figure out where I goofed, and I expect you to write that on your sheet. Okay, so I definitely know you watched these videos. So some of them are me, and some of them are other ones that are good videos as well. So as you can see, they're right there on Canvas, right under the Lab 3 module. And that's all you have to fill out for homework and do for homework next week, y'all, is read the 2 lab exercises, fill out the terms, and watch the videos and make notes on it. The rest of the packet is actually for you in class, but again, you should read over it and become familiarized as to what we're going to be doing. So you notice there's lots of blanks that you'll fill in class that day. And it even says at the top, read. You will do these procedures in lab next week. Right? So make sure you read over, right? And I have a particular protocol, the streak plate, written out for you as well, in addition to what's in your book. And as we progress throughout the semester, you're going to start doing these types of things where you write out the protocol ahead of time, right? You write out the standing protocol for homework from your lab manual. In this case, I wrote out it for you, right, to serve as an example, right, of what you'll be doing in the future. Does that make sense? Okay. So not too bad, right, on the homework front. Which brings me to the dreaded discussion about the book. Okay, so they may or may not came in yesterday. Yolanda, like, a little bit snapped at me yesterday when I asked. I was like, listen, I'll help unpack the boxes. Apparently, I'm not allowed to. So, but those of you guys that did purchase online maybe should get an email today. But I would suggest maybe going over there. Unfortunately, last night a student went over there and they gave her the wrong book. So I really need for you guys to make sure before you open that shrink wrap that it says it's the laboratory manual, right? It should look something like this cover. If you want, I can even post a picture on Canvas for you. It may even say custom on it. Make sure you're not getting one of the microbiology textbooks, right, for the lecture classes. There's 2 different ones even. Makes it even more crazy and chaotic. And those are really expensive, right? So if they give you it, maybe you should just go sell it and use that money to buy another lab manual. Ridiculous. So, you know, make sure that you are getting what you're supposed to get. So the other thing is, too, is I really need you guys to go online and reserve your copies, right? So to encourage you to do so, I've already scanned, but you need to read for next week, right? Because obviously, probably very few of us are going to get our lab manuals this week at the rate they're going. So up here at the top, you'll see I have book 3. That's lab 3, the 2 exercises from your book. I'm new to scanning, all right? One of the pages is out of order, but all the pages are there. Okay? I wasn't going to go scan it all over again. Okay? I don't know what I did wrong. I already scanned 4, but I'm not going to email that to you or give that to you unless you prove to me you've purchased the book and it just hasn't come in yet. Okay? Make sense? So you can show me your receipt, or you can email me your receipt, and I'll email you back Lab 4 if you need it. Okay? Good deal? All right. So that brings us to today's lab, which is Lab 2, which is only on one exercise, but it's a pretty big one. And it's pretty much the only time we're going to talk about eukaryotes, right? That's the title of that lecture. Examination of eukaryotic microorganisms. So there is some terminology, right? I'm not going to go through it. As I did last time, and as I'll do continuously. I already posted, actually right now, this PowerPoint for you guys under Lab 2. You'll see it listed. And then this recording will be posted there as soon as I upload it. So, but one major distinction that I am going to go over is prokaryotes versus eukaryotes, right? And hopefully if you guys have taken lecture class or even a biology class, you should hopefully remember this cell type distinction that we talk about. What do prokaryotes lack? They lack a membrane-bound nucleus, right? So, and then we have our eukaryotes, which U means true. They have a membrane-bound nucleus and other organelles. And the nucleus almost looks like a kernel inside the cells. Karyote actually means kernel, right? And so early scientists saw this, and you will see and have to identify for a lot of the eukaryotes that we look for today, you'll be able to see their nucleus. Not all of them, right? But for quite a few of them, we're going to be looking and seeing their nucleus, and of course we are able to see that structure because they are eukaryotic organisms. Not all of them, though. We talk about classification, right? And so the largest classification schematic that we talk about at the top is domains, right? And there are 3 domains, and of course today we're focusing on just one, eukarya, right? And so the rest of this terminology we're going to talk about as we go throughout, right? But I have it all listed in the front for you so that you have it. So as we said, prokaryotes versus eukaryotes. Pro meaning before, so they don't have a nucleus, and they tend to be teeny tiny. So in this picture, actually taken from somebody's gum line, you see epithelial cells, which are our own cells, and we are eukaryotes, right? Our cells have nucleuses. And so you can see this big pink cell, right, with a large nucleus. And then another cell here. Those of you guys take A and P, right? These are squamous epithelial cells. Oh my God, right? They look like a fried egg. Okay, that's how I remember them. Okay, and all these little purple things are the bacteria. So as you can see, much tinier, right? And going to be teeny tiny when we start looking at them under the microscope. Today, we won't have to deal so much with that oil immersion that I talked about last time. There's a few of them, yeast in particular, that are kind of tiny, that we're going to use oil to be able to see them. And one other really tiny parasite that causes malaria that can actually infect our red blood cells. So we've got to go down really small, right? Really high magnification to be able to see these really small guys. Other than that, we're going to stay around 100 times magnified, and you'll be able to very clearly see these because they're so much larger. After today, you guys are going to live under oil. Remember I said, right, we've got to clean up after ourselves. So I was checking the microscopes, so none of them have oil on it right now. So if there's oil on it after class, remember what happens. You have to stay one of these days and clean everybody's microscopes, right? So clean up after yourself. So this classification scheme, right, grouping organisms based on characteristics, was started back in 1735 by Carl Linnaeus, right? So really long time ago. And my bold is not there. So initially the organisms were classified as plants or animals. We know the world exists of many more things that you can't really put in that animal or plant niche, right? They really don't fit that definition. So then the classification scheme had to become much larger. And it's even larger than the standard that most of us learned when we were in school, right? Of kingdoms, phylums, classes, orders, families, genus, species, right? And then domain, you may or may not been exposed to this much higher classification of for life, for living organisms. What's not listed here that you saw in your reading today? Supergroups, right? Where did those fall? Where do they say that was? It's actually replacing or above kingdoms, right? The good news for you guys is we're not memorizing any of that, right? Because that's all you're doing is filling your brain with something, right? That will eventually change over time. The more important thing is understanding that we have a classification scheme, right? And the reasoning behind it, okay? So we're grouping organisms based on similarities, right? So in the case of this example of going all the way down to a single species of bear, right? In the larger grouping at the genus level, right, you have several different types of bears, right? And at the family level, right, you get the cute little panda bear, right? And as we're going up, right, it's encompassing more and more animals, right? So we get to the kingdom level. So, how are these groups determined? Why is it they put all the bears together? They have similar characteristics, right? Maybe so, right? Well, anyone know what the defining characteristic of the class Mammalia is? It's our class. Yeah, we typically a live birth, hair or fur, milk, right? Hence the mammary, right, glands of mammals, right? We produce milk. Chordata refers to the spinal cord. Carnivores, right? They all eat what? Meat. Even my second graders learning about carnivores and herbivores and omnivores. But nowadays, it's also a combination between traits and sequences from ribosomal RNA, right? So as I said, things are changing as we're learning different things about different organisms, especially when you get down to microscopic organisms. They're not cute and fuzzy, are they? Right? So we're really looking at, really molecular, right down to what are their molecules made of? What type of molecular characteristics do they have? And that's actually how the domain system came about, right? How we ended up with the 3 domains that we have today was because of the work of a scientist looking at ribosomal RNA. And ribosomal RNA are found in ribosomes, right? As well as protein. Every living cell has ribosomes. They have to. You have to be able to make proteins, right? So anything that's a living cell, we can look at this sequence and compare. So remember, today, we're just doing one domain, eukarya, right? Which all of these fall into that category as well. So when I was in school, and even today, a lot of people use mnemonics, right? A device for helping you remember. So for me, it was, Kevin, please close our front gate slowly, for remembering kingdom, phylum, class, order, family, genus, and species. Another colleague of mine, the saying that he remembered was King Philip came over from Germany. But I like mine because I can add the domain at the top. I just changed the sentence to, damn, Kevin, close our front gate slowly, right? And so I've got the domain up there, up at the top, right? So not to be confused with things like pneumonic plague, right, which is caused by a bacteria, right? This is mnemonic with an M. And so taxonomy is classification, right? Grouping organisms into categories. Scientists love to put stuff into groups. Phylogeny, on the other hand, is a little bit different in that we're looking at the evolutionary relationships between organisms, right? Who potentially evolved from who? Who is whose ancestor? Ontology is the study of their development. And sometimes, right, this may or may not be true, ontology and phylogeny may be linked, right? So as an organism develops, it gives you an idea of how it's related to other organisms as you look at the whole developmental process. So how many of you guys know that we have tails? Right? We have a tail. We really do, right? Except for most of us, right, it doesn't continue to grow, right? So we're not like cats and dogs in that aspect. But we are, related to a lot of animals that have tails. Primates, right? Monkeys, they use their tails, right? Theirs develop further. Ours have not. Evolutionary-wise, we don't have the need for it. Okay? And so therefore, most of us don't have one. A long one anyways. So when you get into comparing characteristics too, things can get kind of tricky, right? So when we look at our whales, sharks, and fish, they all live in the water, they all have fins, they all swim, right? But when you look at even just whales and fish, they're quite distinctly different from each other, right? Whales are in fact what? Mammals, right? They have hair, they have live birth, right? They have to actually go to the surface to breathe air. Unlike fish, which have scales, right? And they lay eggs, and they can actually acquire oxygen from the water through their gills. So although, that adaptation to live in that environment makes them relatively similar, right? They have fins, they swim, they live in the water. They're actually quite different from one another. So there are a lot of factors that we take into consideration when we group organisms, right? We can't just look at, one small group like water and fins, right? So phylogeny, right, we're basically trying to draw the family tree for the organisms on Earth, right? Who were their ancestors, right? Just as we can do with people, and this is actually someone's real family tree, right? These two guys down here, or is that a girl? I'm not sure. Bree, D-R-E. Could be a girl. Could be a guy. They don't put symbols on this one. These two individuals right here, right, have a common ancestor, a great-great-grandparent, making them third cousins. Right? So when we say ancestor, like when you say ancestor to for a bacteria, right, or for eukaryotes in general, we say they're possible ancestors, right? So they're grandparents we're talking about, right? Does that put it in a perspective that makes sense to you guys? Hopefully. Maybe so. Okay. All right. So speaking of that, right, you almost can think of cross-mating or interspecies mating and sharing of things. Life forms are pretty interrelated that we look at today. There is a theory, endosymbiotic theory, states that several of the key organelles in eukaryotes. So what's an organelle? What's an organelle? What's the nucleus? It's an organelle, right? It's a membrane-encased compartmentalization within eukaryotes. It holds what? It holds the DNA. Does that mean that prokaryotes don't have DNA? No, it just means it's not encased in a membrane. And in fact, for the nucleus, it's two membranes. Organelles are organization within the cell through the use of membranes. So come on, guys. You remember from biology class, what are some other organelles you'll find in a eukaryote? Mitochondria. What about for plants? Chloroplasts, right? Those, if you look at them, if you look at mitochondria, if you look at chloroplasts, they have their own DNA. They even have their own ribosomes. Those ribosomes are prokaryotic, potentially bacterial in origin. So the belief is that at one time, an early eukaryote engulfed a bacteria. So For chloroplasts, I believe it was what we see today as cyanobacteria. These are photosynthetic bacteria. Right? That organism itself, all by itself, can exist. But when it became engulfed by a eukaryote, they began this symbiotic relationship. And what happens is they start sharing things and taking over duties for each other. It's kind of like when you get married, right? You decide who's going to do the dishes, who's going to do the laundry, right? And then after a while, you kind of forget how to do these things. You're like, oh, I just can't do the laundry. I don't remember how to do that. You're just going to have to keep doing it, right? And that's what happened for these organisms, is that, you know, they lost some of their characteristics to be able to live on their own. That eukaryotic organism became so dependent on that chloroplast for generating energy, it can't live without it. And that chloroplast became so dependent for other things by the eukaryotic cell that it can't live on its own anymore either. But there is definitely direct evidence, the same holds true for mitochondria, that it was once a free-living organism all by itself. Pretty cool, huh? So when we look at the phylogenetic tree, right, this potential relationship, ancestors, who's closely related to who, as I said, Carl Woese and apparently George Fox, who gets forgotten about. You know, if you're the second inventor, you always tend to get dropped. Second contributor, yeah, grad student, forget about it. So he was looking at ribosomal RNA, right? He was looking at that sequence, those G's, C's, U's, A's. He was looking at that sequence and he was comparing them. And he found that he had this divergence of 3 distinct groups. And he named those groups bacteria. So most of the bacteria we're used to being exposed to are in this group. Archaea, which most people kind of a lot of times lumped in with bacteria because they are prokaryotic. They're single-celled organisms. They don't have a nucleus. But come to find out, they're quite distinct from bacteria. And a lot of these guys are what we call our extremists. These are the ones that can live in really extreme environments. In the hot springs in Yellowstone National Park, right? In the Dead Sea, right? And the Great Salt Lake, really salty environments, very cold environments and glaciers, right? Up in Alaska. And then you have eukaryotes, right? The concentration of what we're going to look into further today. And so then, of course, there's us as animals, right? And fungi, right? What do you think of when I say fungi? Mushrooms? Anything else? Yeast? Mold. Mold. We saw a lot of mold on our plates, right? And so, but of course this is microbiology class, so we're not going to talk about animals or plants. We're going to stick to the small things like fungi, right? And ciliates and slime molds and entamoeba. So, of course, we are animals, right? We put ourselves on this little pedestal a lot of times, right? But we are animals, right? We're mammals. We're carnivores. Okay, so then down from domains are these supergroups. And depending on where you're looking in literature, there's either 5 or 6 different groups. So this particular tree that I clipped out from the internet has 6. But our book combines these 2 into this supergroup. So we're going to go through and look at representations of each one of the groups. And again, by no means do I want you guys memorizing the names of these groups. I have difficulty saying them. I usually cheat and have the how do you say on the internet page say them for me. They're pretty phonetic though. Unocutata. It's pretty much the way it's written out. It's how you phonetically say it, if you can pronounce every syllable. So for humans, we are right here in what's called Metrozoa, right, this subgroup. And so there's a really cool website done by Berkeley that I put on the links you'll see in Canvas. There's a lab to web links and videos, right? So definitely check it out. They've done a really good job of all the animals. So as far as domains go, remember which 2 did I say are prokaryotic? bacteria, and archaea. And we're going to live in the bacterial world after this lab. Today, though, we get to be in eukaryotes, which all fall into one domain, obviously named after their cell type, which is eukarya. Now, again, as I said, sometimes we have what's referred to as the kingdom of plants, animals, protists, and fungi. And unofficial groups that you'll sometimes see is protists. And when we're typically talking about organisms like protozoa and algae. Fungi, as you guys have already said, typically we're talking about yeasts and molds. And actually a mushroom is a mold. The mushroom is just the very large fruiting body that's produced by the mold that's living in the soil. So when we get to our first group we're going to look at is excavata, right? And the subgroup we're going to look at has pretty looking organisms, which I wish we had, real ones, because they really move really cool, but they're pathogenic, so play around with those. And you guys reckon this guy from your homework? The Giardia, right? He looks like he has eyes, but what are those? Those are nuclei, right? And then Euglena, I think, is what I'm going to order us some slides of. But they're, of course, much more fun when they're alive, when you get to watch them swimming around, right? So for this supergroup, they are unicellular. They tend to have a feeding groove. They tend to have one or more flagella. And they typically have a life cycle where they have a trophocyte phase and a cyst. What's a trophocyte? Or anyone know what a cyst is? Right. The cyst is the resting phase, so therefore, by contrast or opposite, the trophocyte is the living, feeding phase. Right? So for some of these, we'll have both, because there are periods of time in the environment where they need to kind of go dormant and rest, so they're not killed. So the unfortunate thing about this list is we don't have a single one of these to look at under the microscope today, which is a real bummer. So if we were to test you on any of them, we'd be using the pictures from your book. So for this subgroup, we have Trichomonas vaginaris. Can infect the what? Vagina, hence the vagina-like name in it. So these organisms have a nucleus. You can clearly see that in these pictures. They have flagella, which sometimes are difficult to see. They have an undulating membrane. That's why I wish we had some live ones, because they look like they're dancing. They're kind of cool. You can probably find some YouTubes on them. What isn't on YouTube nowadays, right? They only known to have a trophocyte phase. They haven't found any cyst phase for these individuals. They're fermenters. They don't use the process of oxidative phosphorylation like we do. They're not capable of it. They have hydrosomes, which produce hydrogen gas under anaerobic conditions. Interesting organisms. And they can cause a sexually transmitted disease known as trichosomiasis. So that should have been one of your questions in your book, right? In your homework? Yeah. Not to be confused with trichoniasis or trichinella, right? Or some other tricky. I feel like that's what I'm saying, right? Diseases. Often, but not always, can cause an infection of the vulva, which are sometimes referred to as the lips for females, or also the vagina itself. Males can be carriers, but this organism doesn't really like the male whole reproductive tract thing, right? Prefers female reproductive tract, the vagina, much different environment, different groups of organisms, different pH. So Giardia, can you see the 4 pairs of flagella in this picture that was in your book? No, it's a crappy picture, man. I mean, that is just pitiful. The sad thing is our slides are even worse than that, which is why we don't even bother taking them out. I have years upon years of microscope experience, and so does one of my other colleagues who's a parasitologist. We actually, for fun, one day had a race and tried to find on the slide, right? It took us a while, so I'm not. We don't even bother making you guys. If you want a challenge, right? I'll give you one of these slides, and you can see if you can find it. Good luck with that. Unfortunately, not the best slides. So this is a drawing, right? Has a representation, and again, what really helps you key in on these guys is those paired nuclei, right? They almost look like they have eyes, right? But it is the nucleus, and it's actually split in two. So that's the trophocyte. They do form cysts. They have non-functioning mitochondria, so again, they rely on glycolysis. They have the 4 paired, so a total of 8 flagella.
Will they always have that? Like if you're looking at a slide and you can like count the flagella?
Oh yeah, you're probably not going to be able to, because they're teeny tiny, and you'd have to have really good stain, really good resolution on your microscope. They do form cysts, which are resistant to water treatment, and that's why it can cause infectious disease. It's one of the reasons why we talk about them. Typically, they would be found in stagnant water, right? So sometimes it was referred to as beaver fever, because stagnant ponds that were made by beavers damming up waterways. It is an intestinal parasite, right? So it gets into your intestine. It actually has sucker discs. It sucks and attaches to your mucus layer in your intestines. So it can cause really bad diarrhea, malaise, excessive gas, but it can also be where you don't even know you have the infection. It can be self-limiting and take care of itself. Euglena. These are one of my favorite ones. These guys are cute. So they're green. Why are they green? They're photosynthetic, right? They have a chloroplast, right? They tend to have 1 or 2 flagella, so they swim around and live in an aqueous type environment. Because they do rely and want to be able to utilize light energy, they have an eye spot that helps them be able to head towards light. So they have the flagella so they can swim, the eye spot so they can detect light and head towards it. But when light is not available, they can actually utilize organic sources of carbon to grow, right? They don't necessarily need the light energy. When they use the light energy, they're referred to as autotrophs, and they typically are utilizing carbon dioxide with the help of light to form organic molecules. Or they can just eat other organisms, right, or other food sources that are already made when light is absent. So what structure gives these organisms their photosynthetic ability? Their chloroplasts. And that's what makes them green, the pigments, right, that help them absorb the light. And in this case, things that are green are actually not using green light. And that's why they appear green, because the green light is being reflected back to you. The other wavelengths of light are being absorbed. Trypanosomas. These guys are really cool. As you can see, right, they typically would infect somebody's blood, because you can see red blood cells on this slide. These are little wiggly guys too. They have 1 enormous mitochondria. What disease do they cause? African sleeping sickness. And there's actually another. So notice this is a genus name. They really should have specified what species, because there is another species that causes a different disease. So Trypanosoma cruzi actually causes Chagas disease, but Trypanosoma brucei, and there are 2 different subspecies, Gambesi and Rhodesia. Gambesi is what causes serious sleeping sickness problems for humans. So that brings us to our next subgroup, or supergroup and then subgroups. Our good old green algae, right, which are our chlorophytes. And we won't get to see this organism in this beautiful of a detail. This is one little cell that forms a colony of cells. Does anyone know which one this one is? This one's Volvox. Yeah, these guys are really cool. They live in colonies, in these large colony bundles that you see here. And in fact, this is a colony that's getting disrupted here, and colonies form within. So here is what we would call a daughter colony to this original, and then you have even daughter colonies within this one, right? Little colonies that are forming on the inside that'll eventually pop out and go off on their own. So these are actually granddaughters in here you're looking at, right? Because there was a much larger colony at one time that's breaking up right now in this picture. So we will just see them like this under the microscope for us today, right? And you'll be able to pick out the daughter colonies. We won't be able to see the individual little tiny cells that make up the colony itself. But this is what these guys look like. So as you can see, again, they're green, so they're photosynthesizing. They got an eye spot so they can direct. And then they have the flagella, again, so they can move in the aqueous environment they live in towards the light. So, and they're strung together. They're connected to each other by cytoplasmic connections. I need some water. Chlorophytes, these are pretty cool. So our represented organism in this group is Spirogyra, and this one's very easy to identify because its chloroplasts are in spirals, hence where they get their name. And these guys form filaments. So there's one cell, another cell, another cell. So one of the ones that your book mentions that we don't have is Chlamydomonas. Although a lot of times when we get the amoeba cultures, Chlamydomonas is in there as a food supply. So you'll see these little brown things running around, right, in the fluid around the amoeba. That is Chlamydomonas. And these guys, again, they have chloroplasts. They're autotrophs, so they're using carbon dioxide as their carbon source. And they have cell walls made of cellulose. It's very closely related to what are we thinking of when I say chloroplast and cellulose, cell walls? Plants. Plants, right? And actually what cellulose is glucose, right? That's sugar hooked together. But the way it's hooked together, it's very structurally sound. And the bad news for us is we don't have enzymes to break apart those glucose. And in fact, cows that eat a whole lot of grass, a whole lot of glucose, cellulose, they don't have enzymes either to digest it. The microbes in their gut are actually what are digesting it. Right. So this is what we typically call fiber in our diet, right? It goes in one end and out the other. Helps keep everything doing that too. It's also why you want to chew things like a corn kernel, right? Do you know what that is? What a kernel of corn is? It's a baby corn plant, right? It is one cell. So if you don't bust with your teeth that cell wall layer, that kernel goes in one end and right back out the other. You gotta break it. Otherwise, you don't get the nutrients in the inside. Why do you think cows chew so much? They're trying to break it down for their microbes in their multiple stomachs to be able to digest and get to everything. Although their enzymes can help break it up. Right? So you're actually, your vegetables, you're supposed to chew them, as if you pureed them. So, as far as I'm concerned, just throw it in the blender. So, Chlamydomonas is this pear-shaped, right, algae. It has a chloroplast. It's haploid. What does that mean? Deals with chromosomes. How many sets do they have? One. Haploid is one set. What are we? Diploid, we have 2. Where'd you get your sets? One from mom and one from dad. You got backup copies, I like to call them. Not the case for these guys. They're unicellular, one cell, one organism. They have a chloroplast, right? So they're photosynthetic. They tend to have 2 flagella. They have the stigmata, or the eye spot. Allows them, again, to move towards light. Phototaxis, referring to movement towards light. Taxis, I always remember taxi cabs, right? Take you places. So, although I don't have time to play it, I have it on the list. This guy is hysterical. This is a professor. He literally is singing a song about Chlamydomonas. And Peter found the video for me. I used to just have the song, but it's short. It's funny. It makes people like myself giggle. So that's in Canvas for you guys as well. This guy, Andy Irwin. So again, Chlamydomonas kind of sounds like chlamydia. Not really, right? Don't get them confused. Chlamydia is a bacterial infection. Actually, the #1 sexually transmitted disease out there. And conjunctivitis, right, which is an infection of our eyes. And then #2 sexually transmitted disease is Neisseria gonorrhea, also sometimes referred to as the clap. And you cannot get it from toilet seats. Okay, Volvox. Right, so these guys are cute. I think they're so cool. Right, so they're colonial, right? So it's a whole bunch of little cells that are making up this whole big colonies that you're going to see under the microscope. They can reproduce sexually, right? So this whole colony will be sperm that will bust up and go run and find the eggs. And then a whole nother colony will be nothing but eggs, right? And they'll reproduce. There's a really cool website, too, that I linked you guys to that's short and sweet, where the guy, some other person who's obsessed with Volvox, put together a really cool-looking website where I stole these pictures from. They can reproduce asexually. And so when you look at these, you need to identify a colony and then the daughter colonies within that colony, if it has any. And what are those daughter colonies? What type of reproduction are those? That's the asexual reproduction. So they're exactly the same, right? All of them in there. So there's the cool website, as well as I have it on my links page. Spirogyra, which actually is a band too, named Spirogyra. And as I said, this one's pretty easy to identify because of those spiral-shaped chloroplasts. Haploid as well, so one complete set of chromosomes. They can do conjugation, where they can join up with each other and exchange their genetic information, right, and form what's referred to as a zygote. And that's diploid. And we do have a few slides. They're expensive, right? New slides that I got in that actually show the conjugation on them. So definitely check out the zygospores and share those slides. So you need to, on the regular slides, right, where you could see the chloroplast where conjugation isn't happening, you need to be able to identify the chloroplast, the nucleus, which will be really dark and right in the center of the cells, and then the cell wall that separates the different cells within the filament, which you can clearly see here in the conjugating cells. You can see that cell wall very clearly. So our next supergroup, these are my favorite ones to watch in live culture because they are, most of these guys are motile bicilia and they swim around. You've probably seen this guy before in biology class, right? Who's that? That foot-shaped one. Paramecium, right? This is another one of my favorite ones. I wish we could get some live cultures into this guy. This is a centaur. They can attach and form this trumpet shape, but they can detach too and swim around like Paramecium do. This guy's a ciliated well. You can't see it as that well and actually can form a cyst. This one is pathogenic for us. This is Bellatinium coli. And then here's a ciliate, I forget the name of this one, starts with a B, actually eating some diatoms, right? And diatoms, these guys are photosynthetic. They're not motile, unlike the rest of these guys in this big group. But they have, well, I won't spoil it yet. We'll get to that question. A lot of people call them nature's stained glass, right? Because all the pretty different shapes and the different colors they'll refract depending on the type of microscopy you're using. So, speaking of pretty blooms, right? So this is an algal bloom. These are actually organisms that bioluminesce on their own when they're disturbed, right? They can generate light, and that's why it looks like it's glowing there. So remember I talked about how that chloroplast and the mitochondria came to be? Well, for these groups, especially the photosynthetic ones, there's a belief that, you remember, a pre-algal cell engulfed cyanobacteria. That became the chloroplast, right? Or what's referred to as a plastid. Then they believe for this class, that pre-cell actually engulfed a whole algal cell, right? That already had. And then there was some degradation that happened of that algal cell into the plastid that's found in this class. So there's 2 levels of endosymbiosis that happen, they believe. Kind of interesting. So our representatives of this group, these are heterotrophic or autotrophic. It depends on the environment, right? Are they going to use carbon dioxide, or are they going to use organic sources of carbon? They tend to have small membrane sacs beneath their cytoplasmic membrane. For the ciliates, they all have cilia. They're completely covered in cilia. They have an oral groove to be able to take food in, right? And within that oral groove is the cytosome, which is the tip, basically its mouth, where stuff comes in. So for paramecium, centaur, Bellatinium coli, those are our examples of ciliates. And then we have one non-motile that has a quite complex life cycle, and that's our Plasmodium falciparum. Anyone know what disease that causes? Malaria, right? And then the diatoms are in another subgroup. And most of the ones in that subgroup have a very unique flagella, but diatoms, on the other hand, do not have a flagella. But one of the reasons why we look at them is they're very prevalent in aquatic systems. And they're beautiful. So for our paramecium, right, next week they should get live cultures in, so I'm hoping to bring some live cultures and maybe we can watch them swim around. But we have the prepared slides that you need to be able to identify the nucleus, usually just the macronucleus you can see. We don't have really fancy slides, so you're not going to see the cilia, right, unfortunately. Or the oral groove, right, because it has to be oriented in a particular way and stained in a particular way to be able to see it. This is a scanning electron micrograph, so you can very easily see that groove and you can see its cilia. So about as exciting as it's going to get for us is identifying that it's a paramecium and that it has a nucleus. The contractile vacuoles are pretty awesome. And again, if we get some live specimens in, we may see it. But I've got some really cool YouTube videos that I've linked for you that you can watch these. That's a specialized vacuole that takes in the water and then squirts it back out so the cells don't burst. So they always look like they're winking at you. Kind of cool. Centaurs, as I said, these are really fun to watch swim around. These guys are trumpet-shaped, as I said. They have beaded macronucleuses, so that's these beads that you see along the inside. The cytosome is that white spot you see there in the oral groove. That's, its mouth where stuff is going to enter in. So the cilia, it's covered in cilia, but these cilia are around this top edge, or what are going to move stuff into the groove and then into the cytosome to be able to be ingested by the organism. They're naturally green, although they don't photosynthesize. They're green. Bellatinium coli, again, we have a prepared slide. This is another ciliate, so if we had the ability and good slides, sometimes at high power, high dry, you could focus in and focus up and down, and you can see that it's kind of furry looking. You can see its cilia. How you differentiate this one from the others is it's very characteristic horseshoe-shaped nucleus. Helps in the identification. They are naturally this reddish color. This is the trophocyte, the feeding phase. What symptoms would you have if you ingested this? Yeah, bad diarrhea, right? Bloody mucoid stool. Diarrhea as often as every 20 minutes. And if you have an acute phase of the infection, if you have chronic, it can actually switch between constipation and diarrhea. Thankfully, not really a problem here in the United States, right? More in developing countries like in the Philippines. So only known ciliate, like paramecium, doesn't make us sick, right? Centaurs aren't going to make you sick if you eat them. But this one will. Penicillium coli will make you quite sick. And then we have Plasmodium falciparum. This is non-motile, but so our slides have red blood cells, and depending on how they're stained, right, they may be stained themselves. I can't remember. We got some new slides in, and I think they look red, unlike this picture. But the organisms themselves will be inside the cells or outside, right? And they're teeny tiny. There's actually 2 organisms inside. So what disease again? This is malaria. Thankfully, something we don't have to worry about here in the United States currently. And this is just one species of 4 that can cause this disease, right? There's 4 different Plasmodium. This is the worst one, though. This is the one that's contributed to the most problems and the most deaths. So it's a very complicated life cycle, right? Involves the mosquito, which we certainly do have here. Thankfully, we don't have the Plasmodium. And so you'll see different morphologies, right? And for the different parts of the life cycle. So they're going to be these little purplish looking things with a darker purple looking nuclei on our slides. So any number of these weird morphologies you'll see. It first infects the liver, and then it spreads to the red blood cells, and it multiplies in them and then busts out of them. And that's why it causes damage to both the liver as well as red blood cells. Leads to lots of death. So other mosquito tick-borne illnesses that thankfully we don't have to worry about in the United States. Yellow fever is caused by a virus transmitted by mosquitoes. We did have it here at one time. We don't anymore, thank goodness. Dengue virus, also transmitted by mosquitoes. But certain diseases are transmitted actually by certain species of mosquitoes. The bad news for us is we have all these mosquitoes here. The good news is we don't have malaria, like we don't have wild animal population or humans, large number of humans harboring the Plasmodium. For yellow fever and dengue fever, again, we have got the mosquitoes. We thankfully don't have a large number of the virus to be transmitted. West Nile is a whole nother ball of wax, though. We have the mosquitoes that transmit it, and we do have this virus. And this is what they do test for when they trap mosquitoes. And if you live in an area where they spray for mosquitoes and you notice the trucks coming by a little bit more often than it used to, that means they got a positive result in their traps. You might want to protect yourself a little more. West Nile, for most people, is just going to make you feel like you have the flu. But sometimes it can go neuroinvasive, which means travel to the brain and be deadly. And that's the great fear with that. And then, of course, ticks can be bad news as well, right? They can carry viruses that will attack your brain. And so here's some more information on that kind of stuff if you're interested in it. So, as I said, not really a problem for us here. Major problem in Africa. I'm not going to go over it, but most of you guys are probably familiar with the genetic link between sickle cell and protection against malaria because of the changes it causes in the red blood cells, because of the changes it causes in the hemoglobin that they produce. So that's some FYI information, not anything I'm going to test you on. Right. So because of that, the incidence of sickle cell is quite high in these regions because it's protective against regions where malaria is very high. So then we get to our pretty diatoms, which these aren't quite as pretty as some other ones that you may see. Just depends on how they're prepared. There's two different types that we're going to be looking for on our slides of mixed diatoms. We're going to be looking for the centric forms, the ones that have this circular, spherical symmetry, and then others that have a more elongated symmetry, and those are referred to as pennate, like a pen, or centric, right, circular-like.
Isn't it like a feather?
Yeah. Yep. That too. Yeah. Elongated like a pen or a feather. Yep. These guys have a brownish pigment. They are photosynthetic, right? They are autotrophs. They use carbon dioxide. Unlike the major ones in that subgroup, they don't have flagella. But they're really cool. They have two halves, and the two halves, one of them overlaps the other almost like a petri dish. So if you see, you know, much more powerful pictures of these guys. What inorganic compound is incorporated into their cell walls? Silica. Anyone know what else has silica? That's what glass is made out of. Hence, makes sense, the nickname nature's stained glass. Also known as silicon dioxide. It's a chemical name. So Euglenozoans, our last group, I think, maybe. So we have these guys that don't hold the characteristic shape. This is an entamoeba, and again, unfortunately, the slides we have for these guys forget about finding them. But how did we find them when we raced to find them? This very characteristic nucleus that it has, because it's an amoeba. It's constantly changing shape, right? It's going to be really hard. And it's typically a fecal sample because this is a pathogen. And so you got a whole bunch of other crud you got to try and distinguish between, is this the organism or is this just crud on the slide. Oops, I went the wrong way. So, and then most of us are familiar with amoeba, right? And this is actually a drawing I found on the internet. It was pretty cool, though. So what's he eating? He's eating a paramecium. Yep. Yum. So, Amoeba proteus is the genus that we have. Our slides aren't this pretty. They're pretty much all one color, but you can distinguish and try and find a nucleus. And as you can see, you know, they're stretched out in all these weird directions. And what is that called when they have these extensions of their cytoplasm? Pseudopods. And this is how they move and how they feed, right? They're going to use this to surround. And I've got some links to actually this, to a video I took in this lab, right? When I actually got a good specimen. And we videotaped and watched it move. It's pretty cool. So these are found in fresh water. They're free living, right? We're going to look for their nucleus. No cell wall, otherwise they wouldn't be able to morph like they do, right? So they're continually changing shape by the use of the pseudopods, termed false feet. So a couple of links, and I also have them on that page in Canvas. So Entamoeba histolytica, again, this is a pathogenic one, right? Unfortunately, our slides not up to par. We're not going to mess with them. So this guy will eat you, literally. Its name actually means tissue devourer. It is parasitic, therefore. Cysts are a part of its life cycle as well, so scientists would look for the presence of that in a fecal sample. And again, it's that characteristic bullseye-shaped nucleus, that ring around it, that helps scientists detect the presence of it. Causes what's referred to as amoebic dysentery. And so again, what sample are we taking and looking at to determine if someone has an infection by this organism? We're looking at a stool, right, or fecal sample. We're looking at their poop. So again, usually developmental country issue, can pose problems for homosexual males, travelers, recent immigrants. Institutionalized individuals. Although most of the cases are asymptomatic. What does that mean? No symptoms, right? Person doesn't know that they're infected. So up to about 10% of the world's population is probably infected and don't even know it. Obviously, if they have an acute infection, they're going to go seek help. Okay, so then that brings us to fungi. We leave the fungi for last. I don't know. My humor is bad. Okay, what are these? These are single cells, right? And they're budding. This is an asexual form of reproduction. These are yeast. And these guys are teeny tiny, so this is like high magnification. And then sometimes they'll elongate their cells when they're budding. And so this kind of looks like what you see with mold, what we call the hyphae, the filaments of mold. This is not that, right? This is what we call pseudohyphae. And so for our fungi, as I said, this unofficial grouping that we go by, these guys are non-modal. They have cell walls containing chitin, which is a polysaccharide different from glucose, cellulose. So same thing, still glucose, but linked together. No, wait a minute, this one's not even glucose. This is N-acetyl glucosamine. So it's a similar sugar in that it's a 6-carbon sugar, right? But the way it's linked together allows it to produce that structurally sound cell wall for these organisms. I'm trying to think. Is chitin what's in crawfish and stuff?
Yes,
They're exoskeleton. Arthropods is chitin as well. Yeah, that too. But the thing is fungi isn't really crunchy. I guess because it's so small.
The chitin leads to the flexibility of it.
Uh-huh.
So that's what allows it to become spongy.
Oh, okay.
It depends on what it is. If it's the shells, like if it's the
Yeah.
shell fungi on trees, some of them are.
Oh, yeah.
They're kind of spongy.
Than the others, yeah. So,
That's why they have like the flexibility. But if you snap a mushroom, it's still going to snap.
right. And that's because of this chemical, right, this chitin that makes up their cell walls. So they're absorptive. They actually release enzymes into the environment and then absorb their food, right? So they digest outside of them. So yeast are unicellular fungi. They are vegetatively actively metabolizing. The budding cells are an asexual form of reproduction. As you can see, it's uneven division between the cells, right? It's a little bud that comes off. So there's a picture of, that was the first bud that formed off of this one, and it's already starting a second bud here. So we're going to be looking for that today, right? Trying to see that phenomenon of this budding, this asexual reproduction for our yeasts. And then, as I said, some of them will form longer buds. Their buds become elongated, so they almost look like hyphae, but this is not true hyphae, so it's called pseudohyphae. We do have one of the yeasts that we have, under certain conditions, it produces the pseudohyphae under a certain temperature. I ordered some new slides because some of our slides had gotten broken. And unfortunately, the new slides don't have the pseudohyphae. The older ones do. So we have 2 slides for that yeast for Canada albicans. Broke my thing. I'll just hold it. We're almost done. I need 3 hands. Your molds, on the other hand, are multicellular and filamentous, right? So their cells are elongated. They form filaments. So those individual filaments are referred to as hypha. If you're looking at a whole bunch of filaments under the microscope, they're referred to as hyphae. if you're looking at that whole big mass of the hyphae that we see with our eyes, not with the microscope, so on our plates over there, that's a mycelium, right? That's a huge clustering that you could see with your eyes of all those filaments. Make sense? Okay. So, like we see on our bread, right? But we're going to look at it at the microscopic level. So classification is based on the pattern of sexual spore production and on the presence of cross walls in the hyphae for fungi, which really applies more to the molds than it does, of course, the yeast. So for the yeast, we have two. We have Saccharomyces, and we have the Candida albicans. For the molds, we got three representations, three different genuses. We have Rhizopus, Aspergillus, and Penicillium, which most of us are probably familiar with because we utilize a byproduct produced by that mold called penicillin. So Saccharomyces, right? In your book, I think this, which picture? They have it like this. They're going to look more like what they have is pictured for Candida albicans, right? So they're going to be this purplish, and we're going to have to go all the way to oil immersion to see these guys. So we're looking for the vegetative cells. We're looking for ones with buds. And I think you can see the nuclei in the staining that we have. So I think it looks more similar to this picture I have over here under the microscope. So, does this yeast cause disease in humans? No. Why do we like this guy? It's typically called brewer's yeast, right? It makes beer and wine and bread. Deliciousness. Although I'm limited on the deliciousness now. No more bread. No more bread actually made out of wheat for me. Okay, so for Candida albicans, this one, again, we're going to be looking for the budding, but on the older slide that we have that has the circle cover slip, you'll be able to see the pseudohyphae where the buds have elongated. So I definitely want you guys to try and find that today. And it actually happens when it grows at body temperature. It'll form those pseudohyphae. Is this organism an opportunistic pathogen? Yes, it is. This is what women typically call yeast infections, right? It tends to overgrow in association with the vulva or the vagina. We have it as our natural yeast. The problem is sometimes it overgrows under certain environmental conditions. Things like diabetes, pregnancy, antibiotic use, compromised immune systems can lead to an overgrowth of this yeast and form the condition that we commonly refer to as a yeast infection. If you've been lucky enough to not experience that, the rest of us are jealous. Okay, so then our molds, right? So things that we commonly see growing on our bread. But if we're going to look at it microscopically, right, look down at those individual threads, and then, of course, at their reproductive structures. And the reproductive structures are really going to help us identify one from another, right, where they're producing their spores that go into the air, which is why I said we wouldn't open those plates, right, because then that would release the spores into the air to go hopefully land on some other food. In the case for the mold, for us, some of us inhaling that is bad news because of allergy. So for each one of them, right, these reproductive structures, how they arrange their spores differs. And it'll help us in identifying the three different molds we'll be looking at today. So again, when you look at a mass on an agar plate like here, you're seeing the mycelium, right? It's not until you look under the microscope, and I do have where I've taken from one of these plates and I've stained it with methylene blue. So you can actually see this dark section is where the agar is, and you can see the very thin purple lines, that's the hyphae. And then you could even see the aerial hyphae, the hyphae that's growing up out of the agar in this section. So I have this slide. I'm going to put it out for you guys to take a look at. So for Aspergillus, right, When it grows on those plates, its appearance is going to be greenish to yellow, brown or black, and it tends to have a white edge when you're looking at the mycelium. If you look at it under the microscope, we should be able to identify the hyphae, right, these long filaments. The sporangium, which is this aerial hyphae that's going to give rise to the spores. The spores in this case are arranged in what's referred to as, how do you say it, conidia? Conidia. Okay, I do say it right, conidia. And these are asexual spores typically, although sexual reproduction can happen when two molds come together. It has a different name for the sexual, yeah. So what common commercial products are generated using this genus of organism? Huh? No, not cheese for this guy. Soy paste, soy sauce, right? So these two species are used to ferment soybeans into soy paste and soy sauce. And then Aspergillus nigra actually produces citric acid, right? What we know as vitamin C. And then there's some more info, FYI. So the other one, penicillium, this is my favorite one, other than what it produces. And it's only going to produce this under stressful conditions, right? Because remember, that antibiotic is for it to fight bacteria, to fight for its food source. So on plates, again, this is going to be greenish, powdery, radial floralling colonies, again with another white edge. Underneath the microscope, when you look at its reproductive structures, again, we'll look at the mass of hyphae, but then on the edge, look at the aerial, right, with the conidia. In this case, the conidia are arranged, you see, in these rows, and it almost looks like a paintbrush, and it's actually what penicillium, where that name is derived from. It's the Latin for paintbrush, penicillus. So, again, right, one of the first antibiotics discovered, very useful. We stole their technology. But what food? Cheese. A lot of cheeses are actually made. Those blue cheeses, those blue vein cheeses, it's actually mold in it. And then also used in making sausage and ham. And more of a preservative in that case. Not necessarily for taste. So rhizopus is our one with, has both sexual forms for us to look at. So we have two separate prepared slides. One that has the sporangia. And notice the spore in this case are in this nice cap that you see here. So in that cap are all the little spores that would be released. So that structure that the cap is on, this right here, is the columella? Columella? Tongue twister for me. Right, so these are your asexual spores, and then when two unrelated hyphae come together, they're going to form a specialized spore, the sexual spore, known as the zygospore. This one, to me, kind of looks like, you know those things that fall from the sugar gum trees? The little spiky balls? It's spiky like that. You can really see it really well under the microscope. So on what type of food is this commonly found? You're going to find this, this is one of the main ones that you find on your bread. And I'm thinking I forgot something. Oh, so we have two slides now too. I spent a little bit more money. And this is actually a cross-section of the sporangium that has, you can see the conidia, right, again in rows. But this guy is a ball. So we have other slides that where they didn't slice it, that it looks like this ball. So we have two Aspergillus slides. One where it's whole, and one where it's been cross-sectioned like the picture in your book. Okay? All right, so let's look at some slides.